Question: why is the development of cancer a multi-step process? There are several regulatory checkpoints to prevent a cell from progressing into a cancerous one. Each of these checkpoints is under the influence of many genes for a cell to become cancerous. The genes at each checkpoint have to be mutated such that they are unable to carry out the normal function. More than one somatic mutation is generally needed to produce all the changes characteristic of a full-fledged cancer cell. Multiple gene loci for TSGs and proto-oncogenesis needs to be mutated via loss-of-function mutation and gain-of-function mutations respectively. Thus, the longer we live, the more likely we are to accumulate cancer-causing mutations. Furthermore, since mutant tumor suppressor alleles are usually recessive, mutations must knock out both alleles in a single gene locus before the function of the tumor suppressor is lost. As many gene loci containing TSGs need to be mutated, thus multiple mutations are required. Most oncogenes, on the other hand, behave as dominant alleles, so only one allele in a single gene locus needs to be mutated into an oncogene. Overexpression of the gene, however, this mutation has to be a gain-of-function, not a loss-of-function mutation. Finally, in many malignant tumors, the gene for telomerase is activated. Production of telomerase in cancer cells removes a natural limit on the number of times the cells can divide. More mutations are required for the tumor cells to be malignant, cancerous, and to metastasize. 2 levels. Before malignancy sets in, tumor cells must escape the normal confines of the parent epithelial layer and begin to invade the tissue immediately around it, becoming locally invasive. This requires disruption of mechanisms that normally hold epithelial cells together. Teratoma. The second stage of malignancy involves entry into bloodstream or lymphatic systems, travel via circulation, and colonization of remote sites. This means that cells must cross barriers such as the parent epithelium and a blood vessel epithelium. They may need additional mutations to become able to do this, as normal cells cannot cross the barriers. Thus, metastasis